We're going to get started. Welcome, everybody. Welcome, everybody, to plenary session for the 70th annual TWS Western Section Conference. I've got a couple of announcements before we get started. The members forum, the members town hall will be directly following this plenary. Please come and we'll have awards. We'd like to honor our award winners. Also, it's your chance to participate and find out what all is going on with the section. We also, let's see, we will be sending out a member survey after the conference. Please respond to the member survey. We frequently don't get really great response numbers from that survey, and we need all the input we can get so that we can make the conference better next time. We really appreciate the input. Okay, so our plenary this year was challenge accepted, confront obstacles, create opportunities, and our speakers today all have had some challenges, and they're going to speak to you about how they have met those challenges. And in some cases, they're still meeting difficulties, and in other cases, they have some successes. So I appreciate you giving them your attention. We're going to use a format today where each speaker is going to give a presentation, and we've got cards on your chairs. If you have questions, you can hand your cards out to the edges. We'll have volunteers picking up the cards, and we'll save all the questions for the end. Please, the questions during the session, let's keep them to a little bit more general about the challenges, the overall, how did they solve some problems with their programs, what kind of successes did they have? And if you have individual questions about their presentations or their species, they will be around after the presentation to talk to individual clarifications or questions about their programs, their species specifically. All right, our first speaker is Lisa Shipley. Lisa is a professor at Washington State University, and she's going to speak to you about pygmy rabbit.
Well, today I'd like to tell you a story about an incredible decades-long collaboration aimed at saving from extinction a tiny rabbit that is tied to a declining sagebrush sea. Now, at first glance, nothing could seem more ordinary than a plain brown rabbit surrounded by a bunch of shrubs. Therefore, this story must begin with me first explaining how extraordinary the Columbia Basin pygmy rabbit and its sagebrush habitat really are. So if any rabbit belongs in the Rabbit Hall of Fame, it's the pygmy rabbit. First, pygmy rabbits are the smallest rabbit in the world, weighing in at only 1 pound, which is about half the size of a cottontail and a fraction of a jackrabbit, both species which sometimes share the same habitat. Now, being so small means that these animals require relatively more energy and nutrients per kilogram of body weight. They're not even a kilogram, but whatever. And they also are prey to really just about everything. So pygmy rabbits are also in the Hall of Fame because they are the only mammal in the world that specializes in sagebrush for their diet. In fact, you can see from the light green in the pie chart that that's about all they eat in the winter time. Now, this is quite a feat because sagebrush contains very high levels of plant chemicals that are toxic to about everybody else. However, pygmy rabbits have adapted a unique physiological mechanism that allows them to detoxify these chemicals. And this allows them to take advantage of the abundant and nutritious evergreen leaves of sagebrush year-round. And finally, pygmy rabbits are in the Hall of Fame because they are actually the only rabbit in North America that digs and requires its own burrows. They dig their residential burrow systems usually underneath a big sagebrush plant, which has several different entrances. Now, these residential burrows provide them protection. They can easily escape from predators, and also protection from the very hot and very cold temperatures in the arid shrubland. Now, pregnant pygmy rabbits do something really special. They build a separate natal burrow as a secure hiding place for their neonates. In fact, they give birth right at the entrance of that natal burrow and shove their babies inside, cover them up with soil, and return about once a day to nurse them and recovering them again. And this goes on for the 2 weeks until the young permanently emerge from their natal burrows. Now, these natal burrows are very cryptic, And in fact, no one knew where baby pygmy rabbits were born until the captive breeding program, which I'll talk about a little later, but they've since been found in the wild now that people knew what to look for. So their unique diet and habitat restricts pygmy rabbits to areas within the sagebrush landscape that has deep soils, such as these little mima mounds and drainages. And these are relatively rare on the landscape. Now, with over 400,000 square miles of sagebrush habitat in the Western United States, you would expect pygmy rabbits to be abundant and their populations to be thriving. However, only about 55% of the original sagebrush steppe remains. Over the last century, sagebrush steppe has been converted to other land uses, especially agriculture and some urbanization where that's possible. A lot of the remaining sagebrush steppe has been degraded by poor land use practices, such as heavy grazing, which has caused extensive weed invasion, especially cheatgrass, which has increased fire intervals and fire intensity. And unlike a lot of shrubs, sagebrush does not do well with fire. It kills it. And it can take up to 50 years for sagebrush to return. And even in recent years, we've seen more fragmentation from energy development. As a consequence, sagebrush steppe is considered one of the most imperiled ecosystems in the US. So historically, pygmy rabbits were patchily distributed over 8 states in the Great Basin in the Western US. And fossil records and genetic analysis indicate that the population in Washington, which is that little tiny orange dot on the map, which is now called the Columbia Basin pygmy rabbit, has been geographically isolated from the rest of its range from a minimum of 10,000 to over 50,000 years. Now, Washington Department of Fish and Wildlife began to be concerned about populations of pygmy rabbits in that state in about the 1980s, and has started doing some protective measures for the habitat and the population. But during the 1990s, the Columbia Basin population really started to decline rapidly, as evidenced from annual burrow surveys. And by 2000, a series of fires in the already small populations of pygmy rabbits restricted them to only one small parcel, not small, I guess 3,000 hectare parcel called Sagebrush Flats. And this was a piece of old growth sagebrush owned by Washington Department of Fish and Wildlife. And if you look at the aerial photo on the right, I think it's clear what the cause of the declines has been. In the 1950s, dams were built on the Columbia River, which allowed the remaining deep-soiled sagebrush steppe to be turned into agriculture, which reduced and fragmented the habitat. So by the early 2000s, the burrow counts became so low, the U.S. Fish and Wildlife Service was petitioned and ended up placing the distinct population segment of the Columbia Basin pygmy rabbit on the federal endangered species list. Now, I think most of you know that a distinct population segment, which is often designated based on unique genes or unique habitats, is the smallest division in the ES of a species that's warranted for protection under the ESA. Now, this listing surprised a lot of people, including, I guess, myself, because how could a rabbit species, which are so famous for their incredible ability to reproduce, possibly be in danger of extinction? Well, as it turns out, a quarter of all lagomorphs worldwide are threatened with extinction. And some of you from California might recognize the riparian brush rabbit. And even the common European rabbit, which is from which our domestic rabbits arose, is actually threatened in many parts of its range. So once the U.S. Fish and Wildlife Service lists a species or distinct population segment, the recovery team has to develop strategies to try to restore the population. And there's many different strategies that can be employed depending on the threat and individual factors. But these can include things such as restoring habitat, protecting the population on site, translocation, and captive breeding and release. And I can say that the attempts to recover the Columbia Basin pygmy rabbit have included all of these. Now, because habitat loss was the major cause of the decline in the population of pygmy rabbits to begin with, habitat protection and recovery has been one of the primary strategies, such as taking advantage of federal programs to restore croplands and by purchasing intact sagebrush parcels by the Nature Conservancy and state agencies. In sagebrush flats, the state decided to cancel long-term grazing leases and plow a fire line around it just as a precaution. However, as I said, sagebrush takes a very long time to recover, so conserving and restoring the habitat alone was not going to occur fast enough to save the pygmy rabbit. Now, predation rate is extremely high in wild populations of pygmy rabbits wherever they are. And so federal and state agencies initiated a limited amount of predator control. But as we all know, predator control is not very effective because of leaky borders, constant vigilance, and that it's not very popular with the public. Now, several states had more abundant and more stable populations of pygmy rabbits, so it was logical to consider moving some of those rabbits to Washington to augment and restore the population. However, genetic analysis detected large genetic differences between the Columbia Basin population, even going back a number of decades, and populations from the other states. So because the unique Columbia Basin genome might convey advantages for the unique and specific environment in Washington, translocation was initially rejected as a strategy for restoring the populations, and available resources were directed towards captive breeding. Now, captive breeding, as we all know, have pros and cons, but in this situation, one good thing about captive breeding is that it's when there's no source population for unique locally adapted genes, it can be a good strategy. In addition, in captivity, reproduction, fecundity, and survival can be higher, especially in situations like the pygmy rabbit, where the ongoing threats to its wild habitat had not yet been mitigated. However, captive breeding takes intensive resources, and because of that, there's typically a pretty small founding population, which then leads to problems with inbreeding and low genetic diversity. And some people suggest that captive breeding might create unique selection pressures that result in animals that are a little less adapted for wild conditions. So, weighing all of those things, the recovery team decided to go ahead with captive breeding. And in 2002, the Washington Department of Fish and Wildlife captured the last 16 Columbia Basin pygmy rabbits from sagebrush flats and brought them to Washington State University because we had experience with captive mammals and we had space and resources. These animals turned out to be highly related, all of them on average between a half and a full sibling. And really, unfortunately, no new wild pygmy rabbits, Columbia Basin pygmy rabbits, were ever found in the wild. So within the next few years, captive breeding facilities had been established in 2 other locations at the Oregon Zoo and the Northwest Trek Wild Animal Park. So we didn't know very much about pygmy rabbit biology. They're a species that hardly anyone knew anything about, but our first task was to develop effective husbandry techniques, including housing, feeding, and breeding. So rabbits in general, and pygmy rabbits in particular, are pretty aggressive towards their fellow rabbits. So we had to create individual pens for each rabbit separately, and we needed to make sure we had enough soil so that they could dig their burrows. And we also added artificial burrows and insulated nest boxes, at least as a starter for them. So all of this was a lot of work, and you can kind of tell that a lot of these were handmade by grad students and faculty at WSU. This meant that each tiny rabbit took up a heck of a lot of space and resources. So luckily, we had from some recent master's projects, we did know something about the diet of wild rabbits in sagebrush flats in the Columbia Basin. And so we grew a lot of greenhouses full of sagebrush and collected a lot from the wild. We also grew a lot of fresh grasses and forbs that we knew they would eat. Luckily, these guys did adapt fairly well to commercial rabbit diets, which allowed us to have sort of a base and make sure they got all their vitamins and minerals. So we definitely had no prior knowledge of breeding behavior. And so this was truly a case of adaptive management. very important component of this was that we needed to reduce the inbreeding. So mates were selected carefully based on a genetic management plan to maximize outbreeding. And we learned a lot about breeding, but in the end, we found out the best method was to co-house a male and a female for 1 to 3 nights before they decide to kill each other, per pairing. And females have what's called postpartum estrus. So as soon as they gave birth, they are ready to breed again. So we were able to, at least later on, learned that we could get a lot more breedings in if we bred them then. We also learned that we could tell which animals were pregnant because their fecal pellets became larger, and because of those little natal burrows that we learned about from there when they started building them. So again, maybe we were overly optimistic, but everyone expected these rabbits to literally breed like rabbits. But the captive breeding program faced several difficult challenges even from the beginning. Inbreeding depression resulted in really low pregnancy rates and low juvenile and adult survival among these really closely related founders. About 50% of the juveniles died before they even got out of their natal burrows. And our team of veterinarians from a lot of different places determined that this was mostly due to enteritis, which is basically inflammation of your digestive tract. A lot of it caused by coccidia, which is a ubiquitous parasite that any of you who have livestock know what I'm talking about. Usually doesn't kill your livestock though. Even the adults had problems with disease, especially mycobacteriosis, which is other name is called avian tuberculosis. And it's again all over the place. And it's really only animals and people with low immune systems that have problems with this disease, but it did kill pygmy rabbits. So disease, of course, is exacerbated by keeping animals on soil, but multiple attempts to try to breed them and raise young on more sanitizable surfaces such as concrete were never successful. So to address the inbreeding depression problem, we used a genetic rescue by bringing 2 male pygmy rabbits in from Idaho. And this did kickstart our population and increased reproduction and survival. However, it did reduce the contribution of the Columbia Basin genes to the population. We also started a series of prophylactic medications, alternating different types to try to keep coccidia under control, which I guess helped a little. So over 10 years of continuous captive breeding, we produced almost 900 juvenile pygmy rabbits at Washington State University alone. However, the very high kit mortality in captivity and the natural high mortality of pygmy rabbits in the wild led us to believe that producing enough rabbits in captive breeding population or in captive breeding facilities remained really difficult and very costly. So by 2010, it seemed time to prepare the rabbits that we did have for eventually release. And so the recovery team developed the first reintroduction attempt by practicing with a hard release. In this case, rabbits from the captive breeding facilities were brought directly out and released right into sagebrush flats with a little help from some artificial burrows and a little bit of supplemental food. Most animals from this release disappeared within a few months. Therefore, subsequent releases went on more of a soft release tiered approach. The centerpiece of this was creating really large on-site breeding pens. So right out there at sagebrush flats with sagebrush inside of about 5 to 10 acres. And these were modeled after what was successful in the riparian brush rabbit program. And so for this, what happened was that the adults from the captive breeding facilities were moved into these on-site breeding enclosures and left there. And we could put multiple animals in there. And then any of the young that were produced each year were then trapped out and then either soft released or hard released into the wild. So over time, the recovery team felt like that this population, although there were some animals out there, might needed to be a little bit boosted. And so they started some limited translocation from 4 other states. And although this can boost the population, it did further reduce the contribution of the unique Columbia Basin genes. Now, to examine the initial success of releases, some juveniles were monitored using gluon radio transmitters, but these only last about 2 weeks. So really the bulk of the last 12 years of monitoring has fallen on non-genetic monitoring developed in the Waits lab at University of Idaho. And so each animal that had been in the captive breeding program or in the breeding facilities had genetic tissue punches taken. And so we knew the identity, the ancestry and parentage of all of them. And so now what happens each year, it's happening right now as we speak, large teams of biologists and volunteers walk transects across Sagebrush Flats, hopefully in the snow, and collect fecal pellets from the surface of the snow. And these are monitored and analyzed genetically now in the last few years using genomics. And this helps us get the identity and the contribution of Columbia Basin genes. So you can kind of see a summary of what's happened over the last 12 years. And I'll say even early on, genetic monitoring showed that rabbits could survive and reproduce in the wild, and that Columbia Basin genes could be maintained. And right now we're at about 20% Columbia Basin genome, which is not very much. Also, the animals originally were released in Sagebrush Flats, and you can see those were those green polygons. And then later on, the animals have moved to some restored croplands that were on the edges, and those are the blue polygons. And so the pygmy rabbit population has gone up and has also plunged back down. So to avoid putting all the eggs in one basket, new pre-release pens have been moved to some other sites within the range. And now we know that we have at least 50 unique pygmy rabbits out there. Of course, there's a lot more. That's the minimum number. So 20 years or 25 years of this crazy roller coaster ride has not necessarily ensured that pygmy rabbits will maintain on the landscape. New threats include ongoing challenges such as wildfire, which has burned over 200,000 hectares of the suitable habitat over the last few years, including some of the on-site breeding pens, which has killed some of the rabbits there. And we know that fire is going to increase with global warming. Now, a newer threat is the rabbit hemorrhagic disease, which has recently made its way to North America. And to protect the pygmy rabbits, about 100 of these over the last few years, 100 Washington pygmy rabbits had been vaccinated. So as you can see, a lot of money, effort, and careers have been devoted to saving the Columbia Basin pygmy rabbit. And the question remains, as it does for all endangered species, is it worth it, and why should we care? Well, of course, this is an existential question that all of you have thought about yourselves, but I can say that pygmy rabbits are clearly sentinels of the sensitive sagebrush ecosystem that is sort of the hallmark of the American West. And they're a critical link in a complex food web headed by some vulnerable top predators. And finally, they are ecosystem engineers whose extensive burrowing and browsing has shaped the deep soil sagebrush steppe landscapes over millennia. So in conclusion, I hope you will join Team Pygmy and keep your rabbit ears open so you can follow the continuing efforts to restore this tiny rabbit that is inextricably linked to an ecosystem that currently faces stormy seas.
Thank you, Dr. Shipley. That was great. So we have another speaker coming up, Dr. Callie Crampton. She's going to speak to us from I wrote it down, the Kauai Forest Bird Recovery Project, about some Hawaiian birds that are in peril. And again, hold your questions, put them on the cards, pass them to the outsides. We'll pick them up and ask them all at the end.
No, that's not it.
Good afternoon. Thank you, Randy, and thank you everyone for attending. So I'm going to take you across the Pacific Ocean to the island of Kauai and talk to you about some challenges we have accepted to assure a future for Kauai's endangered birds. And I just want to highlight before we go on that we are at we have shared some similarities, of course, to the challenges that Lisa's described, but we are at different places in that journey that I'll elucidate. And I also want to highlight that I am a field biologist and not involved in the captive breeding program. So when we get to that part of the talk, it will not be as in detail as Lisa explained. And we have a couple people in the audience that I'll direct you to answer questions later about conservation breeding. So to understand what's going on in Hawaii, you have to understand the isolation of the Hawaiian Islands. It's the most isolated archipelago in the whole wide world. And the only things that can arrive on these volcanic islands as they erupt out of the Pacific Ocean are things that take advantage of one of 3 W's. So the first W is wings. They have wings themselves. They're strong flyers. They hitchhike on animals with wings. So plant propagules, for example. The second thing is the waves. They can swim or they can raft on the waves, say a coconut. And then the third thing, which is the story of the Kauai forest birds, is the wind. They get blown off course in a major storm by the wind. And being so far away from where they came from and not being strong flyers, they cannot return. So they have encountered these amazing Hawaiian islands that range from sea level to 14,000 feet. 13 out of 14 terrestrial biomes are represented. Strong leeward effects, windward and leeward effects. So very, 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 very diverse habitats, and they're cut off from ancestral gene flow. There's no more connection with what they once were, so they're kind of free to become something else. So given the 3 W's, only one mammal arrived in the Hawaiian Islands. It's a hoary bat. There's also one that takes advantage of our shores from time to time. It's a monk seal. And also no land reptiles and no amphibians arrived in the Hawaiian Islands. So the birds, the insects, and the plants that arrived in the Hawaiian Islands were free to explode into these terrestrial biomes, unfettered by competition from what mammals and reptiles often do, and also unfettered by predation from those typical predators. And therefore they lost a lot of defenses to predation and other things. So one of the groups that arrived was a rosefinch, a Eurasian rosefinch, that diversified into this group, which we now know as the Hawaiian honeycreepers. So one ancestral rosefinch became over 50 different species, and you can tell from the diversity of beak types shown here that they really took advantage of all those different niches, becoming not just granivores like their ancestor, but nectarivores and insectivores and a wide variety of things. And they lived in the Hawaiian Islands for about 5 million years before humans, the first mammals, arrived about 1,000 years ago, bringing with them more mammals and reptiles and a host of invasive plants, started clearing the lands for agriculture, et cetera, et cetera. So in evolutionary time, these birds have had very little time to adapt to the changes posed by this mammalian and this reptilian invasion. And as a result, we have gone from being the biodiversity capital of the world to being the extinction capital of the world. So here shown on these slides are many of the species already extinct, and the ones with circles around them are on the endangered species list. So a very, very grave problem in Hawaii posed by this, this isolation and this lack of familiarity with modern human threats. And so the culprits are the usual culprits in many island systems. Introduced mammals prey directly on eggs, but also often on females, because the females are naive and don't move off of nests. We have black rats in the islands. They're very, very good predators and climbers on forest bird species. A lot of the mammals also act as competitors for resources. And then the big guy to the right of the rat is a feral swine from Europe, and it's a habitat modifier. It preys directly on seabird eggs, for example, but it also moves through the forest, rototilling and spreading invasive weeds as it goes. It also creates breeding habitat for mosquitoes, which are the next chapter in my story. And then, of course, we've introduced invasive plants like the Himalayan ginger, which take advantage of our year-round growing season to become themselves habitat modifiers and crowd out the food that the birds depend on. And we have outright destroyed habitat by clearing for agriculture and urbanization. So on Kauai, what was once a couple dozen species is now reduced to about 8 species. And I'm going to introduce you to the main players. There are 3 endangered species. The first is a thrush. It's the largest species. It is the only remaining thrush of 2 thrush species. It is 35 to 40 grams. It is a seed disperser. It's the only seed disperser left. And unlike the rest of the species I'm going to describe, it is not a tree nester, it's a cavity nester. And I have a story, if I have time, to tell about a previous program of conservation, breeding, and release. But I really want to focus in this talk, so we'll see how much time I have, but I really want to focus in this talk on the honey creepers and the story that's going on right now. And so the honey creepers is the endangered akekee. It's an insectivore. It was listed in 2010, and its specialty is preying on galls in the ohia canopy. Ohia is the keystone tree species in Hawaiian rainforests with its crossed bill. It numbers about 600 individuals. And the akikiki, another honey creeper insectivore, which preys in the understory on the lichen and the loose bark and mosses, trying to get all the grubs out from underneath it. So those are our 3 endangered species on Kauai. I forgot to mention the population size for the poiohi, or the thrush, is 500. I said the akekee is 600. The akikiki today numbers about 40 individuals in the wild. And then we have one other threatened honey creeper here. This is the EEV. This bird, unlike the other 3, is not endemic to Kauai. It's endemic to the whole state. So it's found statewide, but it numbers only 2,000 birds on Kauai, and it is listed as threatened. The other birds, the 3 colorful yellow birds, are all honey creepers, as I said. So there's the anianiao, the Kauai amakihi. They are declining but not yet listed. There's the red apapane, which is a statewide endemic. And then the little brown guy with his head up like this is the alapaio. And it's a monarch flycatcher. So that's the suite. I want to move now to the story of the honey creepers, the second chapter in our story, and why things are beginning to get so much worse. So in the 1820s, whaling ships introduced the southern house mosquito, Culex quinquefasciatus, to the Hawaiian Islands. And at first, because of the elevational relief that I explained with these big volcanic islands, Culex was restricted to the sea level in the Hawaiian Islands and was not able to climb up our mountains because of the cold temperatures in the middle of our islands in these big volcanoes. As you may know, only female mosquitoes bite, and female Culex mosquitoes in Hawaii transmit avian malaria, which was subsequently introduced to the Hawaiian Islands in the early 1900s. Honey creepers, unfortunately, had no evolutionary contact with avian malaria, and or had once upon a time had it, but in the 5 million years they lived in the Hawaiian Islands with no avian malaria, they had lost resistance to it. So they are very naive to avian malaria and highly susceptible, and many of them will die after a single bite from an infected mosquito. And unfortunately, what is happening right now is that the range of mosquitoes and avian malaria, which are both temperature sensitive organisms, are expanding up the slope of our volcanoes due to warming temperatures and drying weather, which is creating more mosquito breeding habitat. And Kauai, of the main Hawaiian islands other than Oahu, is the lowest elevation island. It's only got about 4,000 meters, or about 1,400 meters worth of elevation, not 14,000 meters worth of feet worth of elevation like the Big Island. So our highest elevation point is 500 feet, or about 1,400 meters. And we know from our mosquito surveillance data that mosquitoes have now moved throughout the range. I don't have a pointer, so I can't show it to you, but they've now moved into the range of the forest birds. So there's a very faint black polygon there that shows you the range of the akikiki, that bird that numbers only 45 individuals. And there are now mosquitoes throughout that part of the range. It's okay. Okay, well, thank you. I'll use it if I need to. So what we have seen as mosquitoes have invaded the upper elevations of Kauai is a concomitant decline in the akikiki and the akaka'a. So the akikiki, like the akaka'a, was listed in 2010, and the reason for this listing was that starting in the early 2000s, we started collecting range-wide data based on variable circular point counts. The picture on the right has some very faint dots on it, which are the survey stations for those variable circular point counts. And then the bigger dots are where the black ones are where akikiki were once detected, and the red ones are where they were detected in 2012. So in 2012, we only had 3 detections of the species. That was 2 years after they were listed. The reason why they were listed is because these data were already showing that downward decline. So in 2012, the population of akikiki was thought to be about 400 individuals, 450 individuals based on those 3 detections. And then the purple and then the blue and then the green polygon show the contracting range over that time period, a couple of decades in akikiki. So similarly, the same data were collected for akaka'a on those survey transects. You can see that akaka'a, when we started, were more widespread and more numerous, but they have undergone a steeper decline since we began collecting these data, and have the 2012 estimate of about 1,000 birds was based on only 12 detections. So in 2013, 2014, 2015, we did a lot of the thought processes that Lisa discussed, and I'm going to go into them again later in the talk because we've had to revisit all those conversations. But in recognition of these steep declines, a population of 400 something akikiki and about 1,000 akaka'a, we decided to implement conservation breeding because of just how fast everything was changing. And because we knew that mosquito-borne disease was the likely culprit, and we had no tools available at our disposal to control mosquitoes on the landscape. And so the result of that, and this is where, as I said, I'll direct you to San Diego Zoo Wildlife Alliance. There's Ron, I want to put your hand up in the back. Maybe you can take some questions if there are any about the conservation breeding. The Fish and Wildlife Service, Kauai Forest Bird Recovery Project, with some help from San Diego Zoo Wildlife Alliance and also Division of Forestry and Wildlife in Hawaii, collected over 4 years 60 akikiki eggs that were then moved to facilities with the San Diego Zoo Wildlife Alliance. And akikiki are, number one, pretty easy to find nests for, and their nests are relatively easy to collect, although we'll talk about that in a second. And they hatch out really well in captivity, and they survive really well in captivity. So those 60 eggs became approximately 45 founders, which was pretty good. Our goal was to have about 60 founders, 45 given the genetic diversity and what we knew about the population. We felt like we'd done a really good job. So we were like, yay, patting ourselves in the back. Now, there are still 45 akikiki in captivity. They are not all founders. There has been breeding of akikiki in captivity. However, it has been just sufficient to offset mortality. So they are not picking up, pumping out babies in captivity. However, they are breeding, and the story of all the things that they have tried to make them breed, and we are beginning to turn that around, I think, and beginning to get to where we have more consistent production of akikiki eggs. But that's where Ron would come into the story and tell you a little bit more about the details that go into that. Akaka'a, on the other hand, their nests are way harder to find. They're way harder to get to. And so we were only able to collect a couple of dozen, maybe 20, 24 eggs to begin with. And they do not hatch out nearly as well in captivity, and they do not make it through fledging to adulthood nearly as well. Plus, I think this year was finally the first year we had an akaka'a chick from those founding populations. So the net result is that reproduction has not been enough to offset mortality, and there are only 7 founding akaka'a at this point in the population, with 1 chick, I think, from this year recruiting as we speak. So one of the things that we're challenges and setbacks, right? So patting ourselves in the back for akikiki, going, maybe we need to think a little bit more and harder about akaka'a, back to the drawing board for akaka'a. But meanwhile, we made some important discoveries in the wild. we were able to band and territory map a whole bunch of akikiki, which becomes relevant for chapter 3 of this story. And monitoring those akike nests, those were the first half dozen akike nests that anyone had ever monitored. We learned that akike do not have regularly 2 egg clutches. In fact, 2 egg clutches are the rarity, whereas 3 and 4 egg clutches are very common. No one had ever documented those kinds of clutch sizes before. And we also noticed that each nest is attended by multiple adults. We don't know the sexes of them because we also have a really difficult time catching akeke, so we don't have very many banded akeke. But going forward and thinking about what you would do for a conservation breeding program for them or a translocation program for them, knowing that each nest is attended by multiple adults is a pretty important discovery. Okay, so chapter 3. So we have all these akikiki in captivity. We think maybe we have 400 akikiki on the landscape because we've done these same surveys in 2018 and the number comes back 400, except we are mapping out those territories and monitoring those color banded akikiki in the core of their range, the site that has the most akikiki. And this is what we are seeing based on our mark recapture data. We are seeing annual survival of adult akikiki, males and females combined, because we don't have that many banded, plummet from 75% to 25% or 30%. I mean, just terrible, terrible survival for adult birds based on 44 individuals. Meanwhile, so this is now 2019, 2020, and we thought we were doing okay, but we're watching this happen. Meanwhile, based on the territory mapping, we are noticing that this site that once had 70 breeding akikiki or 35 pairs plus non-breeding individuals has gone slowly down over time with a slight uptick in 2018 when we did our last survey to 2 and then no birds. So basically akikiki in this time period where we're thinking that we've established an insurance population and we're trying, we're good. We just watched them blink out from this one site in front of our eyes. There is still, or are still, a couple of other small pockets of akikiki on the plateau. There's one by that top northern red dot still, but what we once considered to be the bastion of akikiki is gone. And so we sounded all the alarm bells and we got together with people from USGS and Fish and Wildlife Service and DOFA and others. And we were asked to review in a mediated process all the expert data published and unpublished that was available for those 2 species simultaneously, because the same thing is happening on Maui that I don't have time to talk about. A Maui group did the same thing. So each expert independently considered all the available data and then came up with their prediction for the current population size for each bird and also the year of extinction. So we felt in 2021 that there were probably 45 akikiki left and 638, plus or minus akike left, and that akikiki would go extinct this year. And akike would probably go extinct in 2030 with some margin of error around that, right? So, oh my God, now what, right? So then we revisited all those discussions. We could just do nothing. We could just let it happen. We already have 45 akikiki in captivity, akike number 600 of individuals. Do we just leave them? It hasn't gone very well for the other birds. So hopefully that's not the decision we take. We can move even more akikiki to safeguard them in human care. And so our first effort involved this meticulous egg collection that I don't have a ton of time to get into, but it involved egg collection because removing eggs from the population means that you're not removing all the reproductive potential of adults that have survived to adulthood. Now we are going to remove eggs and adults both. And one of the reasons that this effort is the most highly favored is because it requires the least lead time in terms of permitting and because facilities are already available at the San Diego Zoo, Wildlife Alliance, and other places. So this is a very strong candidate. And the idea is it doesn't even matter if they breed, we just have to get them out of the way of avian malaria and hold them there until we can deal with avian malaria. The next idea that we considered, as with Lisa, was translocation. The permitting timeframe for translocation is a couple of years because this is an extralimitral movement of endangered species to an island that they have never occurred on because they're endemic only to Kauai. So plus it's very expensive, plus we can't even catch akike to begin with, plus are there even enough akikiki to translocate? That idea was shelved. We are working as fast as we can towards mosquito birth control. We are using a bacteria to control mosquitoes on the landscape. It involves a bacteria that when a male and female have two different strains, engenders incompatibility. So the idea is to release tons and tons. These are a male and a female that have the same strain, so they're going to breed. But if you introduce tons and tons of males that have the incompatible strain, the female breeds with them and has no eggs. So we are investigating that to do population crashes for mosquitoes. The time frame for that is 2024, 2025. No time. Akikiki are going extinct this year. So we map it all out, and you can't see the font and it doesn't matter, but it's just mapping the timeline of the year of extinction versus the timeline that it takes to get these management actions on the horizon. And the only thing we can do is bring in Akikiki. So that's where we're at now. I've already discussed how that's going to happen. It's a very, very expensive option, as Lisa has said. Akike'e, however, we are monitoring in place. We think that they will have landscape level mosquito control on the landscape in time for Akike'e. So we're leaving them in place and we're going to monitor them. They're too difficult to move. And then we are managing the threats in the wild. We are dealing with the pigs, we're dealing with the rats, and we're going to deal with the mosquitoes. So if you're interested in helping out, there's lots and lots you can do. Everyone has a role in this process, including addressing climate change with your everyday actions. But this is going to require a ton of resources. So staying abreast of the national and regional discussion and weighing in, please, it all helps. And again, as Lisa has said, it takes a village to make these kinds of projects happen, not to mention all the resources. So it starts with you being here today and listening. So mahalo.
Thank you, Dr. Crampton. All right, again, if you have questions for the panel, we'll do a panel discussion at the end after Dr. Deanna Clifton. It's okay. Dr. Deanna Clifford speak to us from CDFW. And if you'll pass those on the cards to the edges, they'll get collected by volunteers so that we can do a panel discussion at the end.
Great. Well, thanks everyone for staying with us and trying to get over that talk a little bit from Callie, because that's pretty powerful stuff. And I really wish you guys the best. That's amazing situation. I feel like I've had a little bit of a parallel life with Lisa, and so there's going to be some similarities in our talks. But I also want to put in a plug for tomorrow for Joel Sartori coming. So we did have the privilege of having him photograph the Amargosa Vole. So you'll see some previews of his amazing work in this talk. I'm going to talk a little bit, again, more overview, more process, more how we did it, why we made certain decisions with the different species. And I'm going to take you on a trip into the Mojave, which isn't too far away. So our protagonist for this talk is the Amargosa Vole. It is a subspecies of the California vole, quite large, and it has one of its uniquenesses. It has a little beard right here, little white beard that makes it a little bit special. But this is a real habitat specialist. It lives, nests, and burrows in only 3 square bulrush in these marshes that live in the Mojave. That these marshes exist in the Mojave and nowhere else. And this animal is in the Amargosa Basin, which is a very small area southwest of Davis, north of Baker. And you come in through here, here's Death Valley Junction, and there's about a 19-kilometer long area of Amargosa vole habitat in here, but not all of it is habitat for the vole. The vole was once existed in 2 populations, one in Shoshone, California, and one near the town of Tahobas Hot Springs. And I'll show you that in a little bit more. So this is where our vole lives, in these little strips of marshes. It's a beautiful sunset spot. But really, less than 1 kilometer squared is vole habitat, very small disjunct marshes that this vole lives in. And this vole's been around a while though, despite always probably being a little bit rare, it is still there. And interestingly enough, it was discovered way back in the day, and the railroads, you hear about like the borax and the mule train and all the mining in the desert. A railroad was put in that actually vastly altered the Amargosa Basin and started changes that are still in effect now on how the marshes work, how the hydrology of that basin works. And it's bounded, basically the Amargosa River flows through it. It's subterranean for most of that journey, and it pops up in springs and other small places which form these marshes. 100 years ago in the town of Shoshone, the vole was discovered. It was then also discovered in Tecopa, and basically it then went extinct in Shoshone from habitat conversion and the drainage of the marshes, because the marshes have bugs. People don't like bugs. Marsh goes away, water is used for something else. Agriculture comes in, mining comes in, people move in. So we lost a lot of marshes. And then oddly enough, again, another odd human action, a borehole was dug and it tapped an artesian well and inadvertently ended up creating the single marsh that housed the most Amargosa voles for the next 40 years. So, you know, it's strange what happens in these places. The vole was then listed by the department, listed by the Fish and Wildlife Service. And my role came in this little circle here. We're going to focus on that and the two different phases of vole recovery moving forward. So this was a wake-up call. So why is the wildlife veterinarian for the state like sort of running the Amargosa Vole Conservation Program? It's sort of people fall into a certain place at the right time. There's no real logical explanation for it at all. But back in 2010, an enterprising early career biologist, Tammy Varnum, who was out of the Bishop office, got a small grant to look at, just survey the voles. They hadn't been surveyed for 10, 15 years. Where are the voles? Are they still in the marshes? Which marshes are they in? So she started this grant, and then she found these ears. And many of the genitalia also looked like that. And I got a call saying, the ears are rotting off the voles. We don't understand what's happening. And everything looks swollen. They look weird. What's going on? Well, I am not a small mammal disease ecologist, but I know one. So I called Dr. Janet Foley at UC Davis. I said, hey, Janet, look at these ears. You want to go to the desert and see some voles? So we went, basically, and we started examining it. And what this is it's a chigger, Neotrombiculum microti. Right. So here come the vets. We take the, we get the EMs. It's cool. There's, we take some samples of the ears. But the question was, well, okay, some of you have done small mammal work. You see these little orange chiggers on other small mammals and their ears aren't, basically sloughing off like that. So then we got worried. Are Amargosa voles not able to handle this infection? Is there something else about the Amargosa vole or its environment that is making this infection actually cause disease in this species? We didn't know, but we knew that there was a high prevalence in certain seasons. And so we started talking with other people that started working on the vole. And then we just kept getting more questions, right? Does the disease impact the population? What is the survival and recruitment of this vole out here? What's the genetic diversity currently? What are the impacts of predators? How many marshes are occupied, right? It goes on and on and on, right? It's anything out competing voles and what are their real habitat needs? Then we had more partners come in, and that's where we have the Amargosa vole team. So we actually had an early meeting and we just talked about these things with voles and studies started. So Rob Klinger at USGS started a demography study in a few marshes. Davis came in, we started working with Tammy to monitor the health and expand her surveys. And we had a period of sort of intensive data growth. And we had private landowners and local conservancy on board said, hey, we can help. What do you need, right? So it was sort of like a coalition of the interested and the willing. And what we then found out after 3 years of data is that we weren't in a good place. And so this gives you an idea of the vole's current range in the Tecopa Basin. And these red marshes are occupied. Yellow is where sign was observed. The red where we actually did captures. Blue is nothing. And some of these were unsampled. But we went through and these collective groups did this work. And what we discovered is there's a lot less habitat than we thought and a lot fewer voles than we thought. And they're in these tiny pockets of relatively disconnected habitat. And that one marsh where that artesian well was tapped had most of the voles. And everybody else, it was getting pretty sparse. And so this is what ended up getting us into that same space where Lisa has been, where Callie was, is in 2013. We only had one marsh that had more than 20 voles in it. So this became alarm bell time. Absolutely. Now, granted, this is something you often have with endangered species. This is not a beautiful graph. There's not samples every month. There's not people in every marsh, right? It's an incomplete data set, but is it enough to make a decision? And then it was. So this is that famous marsh one where the artesian well is up there. Beautiful, beautiful marsh, 1.8 hectares. Interestingly enough, this marsh occasionally floods the state highway. So not unknowingly and not intentionally, Inyo County roads needed to unclog the tunnel under the highway, which then drained the core marsh that had almost all the known Amargosa voles in it at the time. And by June of 2013, we had 0.17 hectares. And this is literally like a 10-meter strip of habitat that was left. And then what had happened because of the drying is the bulrush actually collapsed upon itself, creating this mat of dead bulrush that was crowding out light and changing the soil conditions in the marsh. So not only did we think we had at least voles there, now we barely had any. And we had to start increasing surveys, and we started making contingency plans. And one of them for us was that we took a surrogate species into captivity, hoping we weren't going to have to take the Amargosa vole, but deciding maybe we should get a little practice. And we could do that without as many permits, and we could do that really quick. So that was one of the first things we did. And then we started to go, are we too late? Do we need to take more drastic action? And then in 2014, it just was not, it was continuing to degrade. So the risk became too great, and we created an emergency captive population at UC Davis, and we increased the intensity of all our conservation efforts. And this was one of our grad students. She actually drew a coloring book for voles, and we have that locally in the system. It's a really cool coloring book, but that's kind of how we felt, right? Like we're on the rescue ship. What do we do? But how did we make that decision? This was the data that we had at the time to make the decision that the time to extinction was predicted to be 5 to 35 years. The 1-year extinction probability, 3 to 30%, again, using the demography study, using the range-wide data we had at the time, and that we were down to that little habitat strip. And there was no way we were going to be able to fix that marsh fast enough. And one of the things I was asked to talk about today was what was the process? And a few people in the room, which I will point out, Scott Osborne, went through this crazy process with us. But the California Department of Fish and Wildlife has a cooperative agreement with the Fish and Wildlife Service where we can implement endangered species recovery actions, and quite a lot of them. So we basically triggered it, and we talked with the BLM, who's a landowner, and we took 20 voles out of Marsh 1 before Marsh 1 was going to get any smaller. And we moved them to UC Davis under that cooperative agreement. But what that set in place because of that agreement is we could only hold them for 45 days. We had to have all approvals in place for a captive breeding program in 45 days. So Dr. Foley and I locked ourselves in a room for 2 weeks and wrote a captive propagation and genetic management plan. And we really didn't leave the room. It was a really intense time. That got fast tracked up through the Fish and Wildlife Service for approval. So our Fish and Wildlife Service colleagues are hustling it on their end, and they had to write a biological opinion, a memo, revise it all, and adjust our recovery permit to allow for captive breeding. But we did it. We did it in 45 days, and we made it like to the day, the countdown for that. And now we're in the ongoing phase where we work iteratively with the Fish and Wildlife Service under our permit, and have our protocols that are all iterative, just like what Lisa had mentioned. Now, I'm going to skip over all the early captive breeding. A couple things. Amargosa voles were nothing like Owens Valley voles. So that was a big surprise to us, but we had animals mostly housed in mated pairs, but we did have some mortality in early litters. We did not have a problem getting them to breed. That wasn't a problem. We had an introduction of a tropical rat mite that came onto the straw that came into the colony. So surprise, surprise, right? But we had fairly well worked out protocols. Where we struggled was getting our voles to releasability. And that was a really big struggle. We grew bulrush in the greenhouses at UC Davis. I mean, we drove back with soil and plants from Tacopa and planted greenhouses, but it was very hard to then recreate mini marshes in a captive setting and make them sustainable. We had none of the ecological processes in place that would sustain these marshes. So the expense was really, really huge. Lots of undergrads, lots of grad students, but we struggled, and we hit personally, like a really terrible event for us, which was our first release. And we had a mortality during the transport of 26 of the 29 animals. So we learned a really hard lesson about some transport conditions. We had brought the 20 back just fine, but we had this the other way. So we had some local heating in the vehicle coming up from the floor. Every time we stopped, it just got a little warmer, and we had 26 lost by the time we got down there. So we actually did the trip in reverse with temperature and CO2 sensors, and we set it up just like there would have been animals in there, to try to troubleshoot the trip and find out what happened. We ended up in a consultation period with the Fish and Wildlife Service, but this is awful, right? We'd had our facility, our captive breeding for 6 months. And I felt like, oh my God, we don't know what we're doing. Like, this is terrible. It was professionally and personally pretty mortifying event for me. But we worked on it. So now we drive overnight. If temperatures are even above 85 degrees at all during the drive gets shifted to be overnight. There's monitors, fans, raised things in the cars, elevated cages. The cage design is different. We changed absolutely everything to make, and we've never had an issue on a transport since, right? So adapt, learn and adapt. That's the rule of the day. Attempt 2, we went back again with 12 voles. We got them into the wild this time, but it was our cameras. It was like Game of Thrones in the marshes. They all started fighting with each other, even though these voles had been kind of acclimated to each other, and nobody ate anything. And basically we had full deaths. We didn't document anybody alive. They had hepatic lipidosis, indicate they literally weren't eating. They were basically had fatty liver, mobilizing all their fat, didn't eat. We re-examined all our protocols and we changed a lot of protocols. Diet change, slower adaptation to the diet, long pre-release period. We only did a 24-hour soft release, changed that, longer soft release. We now pair our captive voles with wild voles because it was clear they did not socialize normally and they just didn't function in a marsh. And we now look at the density of the marshes that we're introducing into more carefully, the placement of our cages more carefully. And the next time we went out, we had almost a 30-day acclimation period with the pairing of a captive and a wild vole. So then people go like, this is really intense, right? We're releasing tiny numbers. The outflow valve is not lots and lots of voles into the landscape. It's small voles, month of release, months of people in the field. Very, very intensive. The other things we saw after a few years of having voles in captivity were changes, right? So again, captivity. When we had originally voles came in and we had them on a diet of rodent chow, supplemented with some bulrush, supplemented with some other vegetables every so often. And then we started to see this condition where they had these abnormal bony protuberances at the molar. It's called molar apical elongation. Well, we'd never seen that in the wild, but years and years of surveillance. We attributed it to 2 things. We changed the diet to rabbit chow, which has more silica and more fiber. And our voles on average are living a lot longer than any vole in the wild. So the 2 risk factors were the diet and the age of the animal. And so we see much less of this now that we have transitioned to rabbit chow, but we occasionally still see it in our very elderly voles. But that's a problem, right? You're not going to release an animal that has that pathology back into the wild. The other thing we saw was change in the gut microbiome. Here's the GI microbiome based on the feces of wild voles, and it shifts after time in captivity, right? So if we put a vole just right back out there, they don't have the same microbiome that their wild counterpart does. And how does that affect their release success? How much time do they need to get back to what a wild vole microbiome actually is? So all these studies are happening in captivity. Meanwhile, right? Captivity is the bridge. The need is the habitat and the water. So while we're doing all this, we're still back in the marshes. And so we're buying ourselves time. And that's exactly what we did. So we give our projects names. This is called the Big Fix. And it's restoring that core marsh. So we put a temporary standpipe in, hand cleared all the dead bulrush rack here, did some fine micro contouring. And you can see the change just by 2016, we're starting to get the habitat back again. This is a vole getting a little tiny VHF collar put on it right there. We had to move the voles because we had to kind of trample the marsh to fix the marsh. So again, a biological opinion. We translocated 47 to other marshes. So we were actually augmenting other marshes. And we did another introduction of founders into the captive colony with 12 more coming. And finally, this tells you how long these things take. 2014, we started that. We finally got the permanent water structure in, permitted and done through all the entities in 2022. We finally have a permanent weir that regulates this marsh. So you have to be in for the long haul. Goal number 2, we want to downlist this animal, but how do we do it if they're all in Tecopa? Establishing a second population. That historical yellow blob now has voles. It is no longer an H, it is a C. And in Shoshone, we have an amazing landowner, Susan Sherrills, and this is her property. And we restored a marsh that used to be a marsh that her great grandfather destroyed. So she feels that it is returning her land to what it used to be. And so this is our sign outside the marsh. What is happening here? The vole is returning home. So this was funded by Partners in Fish and Wildlife and a traditional Section 6 grant. And a lot of volunteers, really, is what it was. And so for 5 years, we watched the marsh, we watched the land, and we made micro adjustments. And in 2020, we introduced the first 6 voles. Didn't have evidence that anybody survived more than 83 days. Kept watching, kept tweaking. 2021, the first 6, and 1 male is still alive today. We also scrapped the radio collars. They were fussy. They took a lot of work. They didn't perform that well. And we now use, we treat the voles like fish. We use biomark arrays, and we lay the line down in the marsh, and they have pit tags. And when they pass through these parts of the marsh, they click, and we detect them via the pit tag arrays. We've really liked those a lot better. And then in 2022, because we still have this 1 male that everybody calls Alfred, we, again, paying attention to the pairing dynamics of voles. We didn't want to flood this marsh with males because we've got 1 in there. We introduced 4 more. And in July, this is a baby vole. So we were really excited by that. We have had 2 litters now of voles, and this is the first time they have been in Shoshone in 100 years. So we are really, really excited about this. We're not sure it's enough habitat perpetually to keep them going forever there, but we're working on more habitat. So again, it's all a process. Really quick, we also did a range-wide assessment, and I can provide that report, and looking at what the voles need across the range. And we determined that, of course, habitat degradation due to water limitation is an issue. We're really paying attention to water depth because that is a predictor of where voles are in the marshes, and that a lot of our marshes are dependent on people. So we have to work with the community in the desert. Absolutely. We have meetings just like, right? Where are we? Where have we been? How do we keep going? So about every 5 to 6 years, our team prioritizes where we need to keep going and how we need to change plans. And that's exactly what we have done. With the recovery plan has been amended in 2019. And we made a big decision last year, and this is sort of where I'll finish, is that we are drawing down our captive breeding program. For some people, this seems like really scary. We think we can do it. And we want to put the money into the conservation. We've piloted new techniques. We think we know how to do it in the wild. And this is a lot of money to keep going. With some benefits, we've had huge information gains in bull biology. We've learned how to release and do these translocations now. And we've had an insurance colony. But we're at the place with some of our habitat restoration that we feel like we can decommission the bridge without a lot of risk. And if we have to bring them in again, we're this much further ahead of where we were 10 years ago. And so that's where we are now. More challenges are being accepted. We're working on community composition dynamics, drought and marsh drying, and starting on a project called the Mega Marsh, because we like big and mega. It makes us feel, it's a vole. You gotta like stamp it up a little bit. And that's another core marsh in Tecopa, so that we have 2 large populations, but it's probably got about a 10-year horizon on it as well. And that's it. I'll be around if you have any questions.
All right, thank you so much. Now we're going to go into a little bit of a less formal discussion where the panel is going to address some of the questions that you guys have come up with. And we can have a little bit of back and forth. And I'm going to have John ask those questions. John Prine, thank you.
And so the first question that's come up, and Deanna already addressed this a little bit specifically, but maybe the other speakers could also talk to it, is dealing with these issues of funding, how much these sorts of recovery cost, but also the need for the continuity of funding, because any break could be catastrophic.
So in our case, in Hawaii, maybe the second question first, the continuity part, you just kind of have to jump in with a leap of faith that when you're doing the right work and they see that it's working, they're going to keep funding, because all of our grants are annual, but everyone knows that it's not annual. The problem isn't annual. And so the initial collection of the Akikiki and Akike eggs in 2015 and 2018 was founded by our local Fish and Wildlife Office primarily. And then to round it out, I was able to attract other, Diana's mentioned Section 6 funding. Section 6 is money offered up under the Endangered Species Act for this kind of work. But it is lots of money. I think that first 4 years was $750 or $1 million in 2015 dollars, and it would cost a lot more again. And I'm only talking about the piece of getting them into Ron's facility. I'm not even talking about another several $1 million that Ron spends keeping them going there and trying to get it. I mean, that is why I think Deanna's talking about conservation, but breeding being the bridge. You really never want animals to be there forever. It's a temporary solution while you wait to fix the habitat and get what animals out where they belong, ideally in the wild. So nowadays, it's mostly we rely on endangered species funding. That being said, doing some non-traditional fundraising has also been very important for us to kind of cover the gaps, and including selling t-shirts, like we do it all to make sure that these animals have a future.
Oh, yeah, that's on. I'll just make I just wanted to completely agree with Callie. Your year to year, which is why it's really good to have both your short term and then your horizon planned out. Because funding fatigue is real, even if, and you feel a lot of pressure that every year you're having to show some modicum of progress. So when you have some dark years, like when we had 2 years where our releases weren't going well, I mean, those are real stressors where you go, like, are we going to still have money? Are they going to, do people still have confidence that we can do this? And the agencies have been really supportive. And so we have bounced from money from Section 6, BLM regional, BLM national, Fish and Wildlife Service regional, crowdfunding by grad students, anything you can get. But I would say, as the co-leads of the research and conservation arm of Voles, Dr. Foley and I, 80% of our conversations always start with funding and who's on what and what grant we're transitioning to and when does this one close and is there a proposal coming. So you do feel like you're building a 30-year program on little short-term chunks of money, which is always precarious, but it seems to be how our system is designed.
Okay, I'm gonna get over here so I can see everybody. One of the questions that we've gotten has to do with just how much all this costs compared to the likelihood or probability of success, and the limited availability of conservation funds. And so a triage question: at what point do we decide to let something go and allocate resources elsewhere, which may have a greater probability of success? Or related to this, how do we prevent getting to this emergency room medicine situation in the first place, even for the species that we feel like we can do something for?
Well, I can probably take the second part. You don't want to wait till there's only 16 rabbits left before you decide to do anything with them. So it's really going to take watching all these species that we're worried about, or even species we didn't know much about, and making sure that we're doing those surveys and keeping track of it. I know that's hard too. Population estimates are really tough in wildlife. And so we've got to plan way ahead of time.
I mean, we have had those conversations in Hawaii with so many species on the Endangered Species Act. And I'm not sure how many of you are aware, but until very recently, the funding formulae disadvantaged Hawaii because a lot of the federal funding for endangered species is based on human population size and the number of borders you share with other states. Well, guess what that does for Hawaii, right? When we have so many species to fund. So, and I think for the longest time, we were watching the monitoring data, and it not only is monitoring data in general hard to get, the rarer a species get, the harder it gets to get the data and the more imperfect your data are. But you know, we were watching those trajectories when we first listed Akikiki and Akake going, we know that mosquitoes are the problem. We know that mosquitoes are the problem. Are we going to be able to control mosquitoes on time? Are we going to be able to control mosquitoes on time? And kept, you know, hoping and hoping. And then finally you just have to be like, no. And we have an obligation, we have a legal obligation under the Endangered Species Act, but we have a moral obligation, especially in Hawaii, where these species have tremendous cultural importance and they have ecosystem. I mean, I didn't get a chance to speak to the ecosystem importance of these species, but we like to say that the rain follows the forest. Without these species maintaining our forests, doing their insect control or functions or seed dispersal functions or pollination functions, we have no forest, we have no Hawaii. So, and you never know which, when you're talking about triage, you never know which is going to be the one that causes everything to unravel if you let it go. So I think we just feel like we have to make it happen and we have to find the ways to make it happen. And luckily we are now a couple of years away, speaking of funding, from what will be the most expensive conservation program in Hawaii, which is suppressing those mosquitoes. And it is looking down, I mean, it is, we will be in that for the long haul. And luckily we have people in high places that are looking for funding in perpetuity kind of, or at least the 10-year horizon we need for that. I'm not sure though, I mean, we have had the conversation, but luckily we've never quite got to the place where we had to be like, let's let that one go. In my lifespan in Hawaii, I think there have been times when we didn't make decisions in time and species did go. That was all those X's on that poster.
I don't really have much to add. I do think each species has its inherent value. We've lost a couple of the desert endemic vole species, and we have a chance and we have momentum to not lose the Amargosa. And I think that's part of the reason why that program continues, but we're cognizant of it, right? The captive breeding portion of the budget takes away from other things that we probably could speed up to have more effective conservation in situ, which is why we were, as some people might perceive, fast to say we're going to scale down the captive program. But part of that is a cognizance that other species need resources. And we think if we protect these marshes that the voles are in, we're also protecting other species that are in that system. And, you know, it's harder for people to wrap their heads around the non-mammalian species we're protecting in the marsh. So if the vole can kind of be the poster child for that, but I don't know, it's a tough decision, right? Like, you know, I mean, I'm looking at Ron in the back because, you know, what would happen if we had let go of the last few condors, right? Like you think about certain species and the vole is not a condor, but it's the same. And I think we'll fight until we can fight. But being proactive earlier, I call it the conservation gray zone and not the red zone. If we can do more interventions in the gray zone when species are struggling versus the red zone when we're already, our backs up against the wall, that's the preferred way to do it. We just don't necessarily have a regulatory and a funding system that allows us to do that.
Also, you know, when you're starting, you don't know what's going to happen. I mean, we really thought we were going to captive breed for 3 years, right? So the problem is once you've put all that effort and all that money in, then, you know, do you want to let that investment go? So that's tricky, right? To start it is the first question, but to end it is another. And after you put the effort in, it's hard.
So we're out here in the West where there's obviously a lot of public land, but you can't do it all on public land. And so can you talk just briefly about the role, as Deanna alluded to this in her talk, but the role of private citizens, of private lands, what incentives they may have, positive or negative, to be collaborators in these recovery projects?
Well, I can say that, you know, Eastern Washington in ranching country is not a very popular place to be concerned about an endangered rabbit. And it was really hard, had to get a lot of different kinds of relationships with the landowners, which U.S. Fish and Wildlife Service and Washington Department of Fish and Wildlife had to do. And it was really tough. They had death threats and everything. The good news is that over the years, I don't think it's as big a deal. It was really big at the time. People were worried that people would take their land away if this was an endangered species. But we've also had private landowners, I think the same, who've donated their land for pygmy rabbits and to Nature Conservancy, and have been part of all of those volunteer operations to do the genetic monitoring. But it was really, really tough. And I think the private landowners and private people are very afraid of the Endangered Species Act because they are really afraid of what that might mean for their private property. But, you know, it just has to start and everybody has to work hard at it.
Yeah, I would agree. I mean, we're in an extraordinary situation in the Amargosa with a passionate landowner, but you know, but it also has to be business savvy for her. And what we're hoping is in Shoshone, her business is going really well. And in fact, all the restoration that she's done, people are coming there more because her facilities, everything looks more lush. There's this beautiful marsh. There's Shoshone pupfish back from the brink. She has the only probably marsh in town that has 2 formerly extinct species in it now, you know, thriving in a very small habitat. But we did it in a way where people can access it. There's education, there's a walking path. And so trying to make those private landowner relationships win-win, making coloring books and Vole stickers that go into COPA so that the little shops that sell something have something with an endangered species to sell on it, and having those community meetings. A lot of our land is federally protected, which is really lucky, some owned by the department. But the thing we found out that's the trickiest for us is that a lot of the water is coming from private lands. And if we have a hot springs business turn off its outflow pipe, we could lose a marsh. So that's where water management now is a new area that we're coming into and talking with landowners. So it's not that they necessarily own the marsh, but they have the water.
So with that, we are at the end of our time, and it's time for the members' town hall. But thank you so much for your attention. Thank the speakers for bringing their projects out and just sort of giving us a little glimpse into some of these challenges that they have faced with their projects. And I hope that that's helped some of you with any challenges that you might face on projects that you're working on or future projects that you may end up on. Resources, there are resources all through the Wildlife Society for those kinds of challenges that you might face. So thank you so much. And with that, I think we're going to call it the end of our plenary session. Thank you.